All right, peeps, I'm going to make this kind of quick because I'm super exhausted. This one goes into the organelles necessary for processing energy and matter, uh, the parts of the cell necessary for processing energy and matter. This first little part just reminds you, hey, if you're going to be considered living, these are the things you must be able to do, right? Genetic material, process, use energy, homeostasis, so on and so forth. So what are the main ones that we're talking about? Obviously, chloroplast and mitochondria, but those are not the only things. It's not just food in the sun. There are a few exceptions, methanogens, hydrothermal vents, that kind of stuff. They feed on methane gas or sulfur gas. And, and of course, we're talking about ATP. So, what are the things that are involved in processing energy and matter? The first one let's look at is lysosomes. Remember, they're the ones that are a double membrane that come from the Golgi. Therefore, they are part of the endomembrane system. The lysosomes, inside of their double membrane, they have hydrolytic enzymes. Therefore, these enzymes, their job is to break things down through hydrolysis. Now, to be able to do that, they've got to release a lot of hydrogen ions. So, the more hydrogen ions you have, the more acidic the environment becomes. So, inside of your lysosomes is a highly acidic environment, which is definitely contrasting to the cytoplasm, the cytosol environment surrounding the lysosome, which has pretty much a neutral pH. So if a lysosome were to burst, no bueno for your cells are going to die. Sometimes that's actually necessary, and I'll get to that in just a second. All right, so in the picture, real quick, just want to point out a couple of things. So you have the lysosome in purple, and you have this thing that came in. Excuse that. No, go away. Little preview window. Don't want you. There you go. That little food vacuole, in this case, it's a food vacuole over here. It's like taking in peroxisome and mitochondria, because those were left over. You don't want them. And lysosome's job is to fuse with vesicles that come in from outside, and when they fuse, they release those hydrolytic enzymes and break it down. Now, what I need to point out, go ahead and see if I can draw real quick on here. Is it going to let me draw? No, it's not. So annoying. Nope, not letting me draw. Okay, that's all right. Go down here, see if it'll let me draw here. All right. So what ends up happening is you have this vesicle that begins to fuse from the outside, and you had a lysosome that was already on the inside of the cell. This vesicle that fuses, we call a phagosome. Okay? Phage for eating, okay, and then some for containment. So it's an eating vesicle. We've contained this food that we ate, and then you're going to see that they fuse. When they fuse, we call that fusion of a lysosome and phagosome a secondary, let's see if I can get that on there, lysosome. Okay, secondary lysosome, because it is now actually the lysosome fused with it, and it's breaking it down, doing its job, its function like it's supposed to. So, all right. Go ahead and just continue real quick. All right, don't know why it's doing, okay, there you go, sorry. All right. Main function, digestion, right? Apoptosis. Apoptosis is programmed cell death. It's a supposed to happen thing. When it doesn't happen, that's when you come out looking a little bit more alien-like than you probably were supposed to, because when you're developing inside the womb, you actually have a lot of extra skin than you're supposed to, kind of like literally surround your whole body. And so apoptosis is supposed to happen, which means the lysosomes burst open, releasing their hydrolytic enzymes and literally killing the cells from the inside out, helping to get rid of, extra skin so you don't walk around looking all kinds of weird and alien-like. When things are bursting, when cells have been lysed, they kind of look like that weird, crazy dead cell that we actually call a bleb. It's kind of weird, but they just absorb all the other cells and just continue to go through apoptosis, such as getting rid of all the webbing between your fingers and your toes. Okay, so all that extra skin and why you actually have, holes in your nostrils and holes in your ear canals, all of that is because of apoptosis. That's a good thing. So yay for lysosomes. Now, lysosomes, sometimes they don't work like they're supposed to. And if the digestive enzymes don't work, that's genetic diseases, and that can actually lead to death, because what happens now is they pick up the stuff, but they can't actually break them down. And so the lysosomes just basically continue to fill up with all this waste and trash, and your cells become this big, giant heap, and they can't function like they're supposed to. So if a cell can't function, eventually the entire tissue can't function, and then the organ, and then eventually you get entire breakdown of the system. One of the most notable diseases is called Tay-Sachs. It's found in Jewish descent, and we'll talk about this when we look at genetic diseases. This is just one example I want you to know. In individuals with Tay-Sachs, again, they come from a Jewish descent. It primarily, you see it staying Jewish descent because, well, Jewish individuals typically marry within Jewish religion. It just keeps getting passed down. It's a recessive disease, but their lysosomes can't break down, so they end up collecting high amounts of fat, and it starts to interfere with neurological process. So at about a year old, when an infant should be beginning to walk, they are still crawling. And then their crawling slows, and then eventually they can't crawl, and then they can't roll. And when they would be able to feed themselves, now they can't. And just you get this slow progression over a couple of years of them going back to infancy, basically, until they can't do anything, even open their eyes, and then eventually they can't breathe anymore. The oldest living individual was 6 years old with this disease. Another example of a some, so something that is a sac that holds on to things, would be a peroxisome. Peroxisomes, their job is kind of what you see in the name. It helps to break down hydrogen peroxide, because inside of this lysosome, in a way, is its enzyme catalase. So it's still hydrolytic, still going to do a job of breaking down. It's just specified. Helps to break down primarily hydrogen peroxide, can break down fatty acids and sometimes sugars, just depending on what kind of cell you find it in. You're going to find peroxisomes in animal cells and plant cells, both alike. You find them in us because we want to break down hydrogen peroxide, because it's actually a byproduct in most of our chemical reactions. And hydrogen peroxide is toxic to our cells. It's not toxic to you as an individual, but inside our cells, it interferes, and it can actually break down certain components. And one of the most notable is gray hair. It breaks down the pigment in your cells. Therefore, you have no more pigment and you see gray hair. Can't be reversed. I'm sorry. You can slow the process. Why a plant cell has a high amount of peroxisomes is kind of odd still sometimes, because yes, they will have a buildup of a little bit of hydrogen peroxide, but not like an animal cell. But honestly, sometimes there's a lot of peroxisomes in a plant cell, more than there really should be. So it's a little baffling sometimes to biologists. Why does a plant cell need all this extra peroxisome? Well, it's because they're probably there for growth purposes, and so that excess sugar or fatty acids aren't, getting in the way of the growth of the plant over time. All right, vacuoles. They are not necessarily a double membraned organelle by any means, but their job is to still store. They can store water, they can store food storage, undigested food. It just kind of depends on what cell you find it in. If you're talking about protists, a protist lives in pond water, surrounded by really fresh water, which is low salt concentration. So Unfortunately, a protist has higher salt concentration, so water wants to rush inside of them. So to keep them from exploding because they don't have a cell wall, they've got to constantly pump out their water using what's called a contractile vacuole. So you actually see this contractile vacuole literally move in and move out, getting the water out of their cell and pushing it back to the surroundings. Plants have central vacuoles, really large compared to the rest of the cell. Why? Because, hello, a plant needs to hold on to a lot of water. Go figure that one. But it can also hold on to like pigments and proteins and stuff like that. All right. We have vacuoles in an animal cell. For a long time, they didn't think we did. But come to find out, yeah, we do. They're just really, really small. We don't really need them, to hold on to water or proteins and stuff. But we still have a few. Mitochondria powers the cell. Woohoo! We're going to go into depth into mitochondria and its process. All I need you to know right now is how to draw it and basically label it. This is right now just structure and basic function. So if you could tell me that it produces energy, I'm good. But you've got to be able to draw this. The fact that it's a double membrane, what it's telling you is this outer membrane never moves. That is, remember, it's part of our endomembrane system, so it has 2 layers. So that doesn't move. But this inner layer actually is folding in on itself, creating what we call a cristae. Inside is a cytosol with ribosomes everywhere and one piece of circular DNA. What organism has circular DNA? Ding, ding, ding. Bacteria. This is why we consider the possibility that mitochondria and chloroplast actually once were their own living organism that we absorbed over time. So be able to tell me a cristae, the open space called a matrix. Function. Here's one thing I need you to be able to get to. Notice I say that cellular respiration is digestion, also known as catabolism, because you've got to be able to associate all these terms together. Cellular respiration is the breakdown of food you eat, which is a catabolic reaction, and that happens in the mitochondria. So once you start being able to use these terms across the board, it can kind of start to pull things together. All right, for mitochondria, those are just really pretty pictures. Ooh, aren't they pretty? Mitochondria, chloroplasts. Chloroplasts are similar in the fact that they have an outer membrane. Okay, they have this inner membrane, and then they have these photosynthetic structures called thylakoids. Okay, again, because they're double membraned, they have their own ribosomes, they have their own circular DNA. Ding, ding, ding. They were once prokaryotes, is our suggestion. The opposite of respiration is photosynthesis, so it's not catabolic, but it's anabolic. Why? Because you're building up a plant cell wall with the production of sugar that comes from photosynthesis. Oh, look at all those pretty photosynthetic cells. Aren't they pretty? All right. The last thing is, what's this theory? The theory is called endosymbiosis, where mitochondria and chloroplast were once their own living organism. Our early eukaryotic cells ate them, like engulfed them, but rather than breaking them down over time, they just kind of became part of ourselves, lost their individuality, and they've remained inside of our cells over time. Why do we think this? They both can make their own energy. They both have double membranes. They both have their own DNA. They have their own ribosomes. They can make their own protein. And they can replicate separate from the cell. Things that our other organelles can't do. So it's just super unique. So be able to tell me what endosymbiosis means, the engulfment of early prokaryotes that eventually became our mitochondria and chloroplast. Again, it's a theory, but man, that's really one well-supported theory, right? This is extra. I don't need you to know this, like specific by any means. Chloroplast, the only thing is chloroplasts are not the only storage kind of grouping inside of a plant. There's all these other guys too that do some really specific stuff. So just know that if I say the word plastid, I'm talking about anything that can create or store something inside of a plant. And the one we focus on is chloroplast for pigments. And that's just extra. All right. So there you go. Talk to you later.